When I was a little girl, I got to hold a starfish. And as I held this thing in my hand, I was so amazed that something so alien, but so beautiful, shared this world with a little 5th grade girl from Michigan. And I decided right then that I wanted to be a biologist. When I was a bit older, I went to the beach with my family, and there were thousands of alewives just littering the shoreline from a recent fish kill. That was my first real introduction to biology and ecosystem issues. We also used to swim at the lake by my grandparents' house. And my sister and I would put clams in this little blow-up boat filled with water and wait for them to open. And then when you pick them up, they shut so fast that they squirt out this little jet of water, and they were like our own living squirt guns. And then we began cutting our feet on giant black beds of zebra mussels. Thanks to the zebra mussels, what was once a fairly healthy lake became open to other invasions, and the clam population declined to the point of local extinction. That was my first introduction to invasive species and how they impact our surroundings. Our lake was changed forever, and I was so mad. I would say, why can't we just get rid of the zebra mussels? Why can't we just stop them from getting here in the first place? But I've since learned that it's not that simple, and it is extremely frustrating. So I grew up, and I went to college, and I received degrees in biology and marine biology from my starfish friend. But I hadn't really thought past that point. I had to decide where to go from there. Questions had begun to pop up in mainstream media, questions like, why are bats and bees dying off? And how did zebra mussels and sea lampreys get here in the first place, and how do we control them? And I thought back to the alewife fish kill and the zebra mussel beds from my childhood, and the more recent reports of Asian carp getting ever closer to the Great Lakes, and just how much I hate invasive species. So I asked myself, is there something I can do? Well, I've got some training now. I could conduct research to help us improve how we monitor and manage these invasive species. Awesome, let's do it. Well, I had absolutely no idea how to even begin to accomplish this goal. So what do? I go to grad school. And it was in grad school that I was introduced to genetic research. And I quickly discovered that genetics will change everything. With genetic technology, we can take something with as much information as hundreds of textbooks, but so small that a full set exists in every single one of our cells. And we can turn it into something visible, that we can analyze and read patterns to gain useful information and answer any number of questions. How might a housing development affect the local coyote population, and as a result, the safety of the new residents and their pets? Why is my loved one sick? Is the water in my well safe to drink? These are just a few questions that can be answered in part using genetic technologies. So, quick science lesson. What I'm showing you here is an image of DNA. Now, DNA shows up as bands of light, and we can cut DNA into pieces and separate them out by size, with the largest bands at the top and the smallest bands at the bottom. So what you're looking at is DNA cut into pieces and separated out by size. Now, each column has DNA from a different species. DNA from different species cuts in different places, resulting in a variety of what we call banding patterns. And different species also have pieces of DNA that are unique to that species, resulting in a unique banding pattern. So in this sense, we can think of these banding patterns as being like barcodes on products at a grocery store. And we can use these barcodes to help us tell apart closely related species and help us figure out where they came from and how they evolved. It's super useful. In fact, there's an organization out there called the Barcode of Life, whose major purpose is to collect DNA barcodes from any and all species on Earth to help researchers continue to push forward with research without having to start over with every new project. Trust me, it's hard. So I was in a lab that worked with water milfoil, which is a type of seaweed. And water milfoil as a group includes Eurasian water milfoil, which has invaded the United States, northern water milfoil, which is native to the United States, and then a harmful hybrid between the two. Water milfoil, it's not just a seaweed anymore, is it? Now, we want to keep the native. We want to get rid of the invasive and the hybrid, but it's very difficult to tell the difference between them just by looking at them. And the most common characteristic to use to tell them apart is to look at the leaves. So here I have leaves from northern water milfoil as an example, hybrid water milfoil, and Eurasian water milfoil. Now, look at all three sets together. I don't know about you, but I would not be able to tell that there are three different plants here, let alone which leaf belongs to which plant. In fact, at conference presentations, I often show this image one leaf at a time to professionals who work with these plants every single day to test their ability to identify them, and you would be amazed at how many get the answers wrong. So in order for my lab to study the ecology and control of water milfoil, we developed a DNA barcode method to distinguish between them. Well, soon other people learned that we could do this, and they requested that our lab identify their water milfoil as well. And that's when we began to run a genetic identification service using these DNA barcodes as a small service out of the university. But I wondered, what if we could do something more? What if this could become something bigger and more practically applied? So I developed the genetic identification service into a full service for citizens and environmental managers, and I established GenPass LLC. GenPass provides genetic identifications of water milfoil to aid citizens and managers in the development and evaluation of control strategies for these plants. This is now one of the very few companies that provides services based in genetics for anyone and everyone, just like companies like Ancestry.com or paternity testing facilities do. Now, I also serve another role as sort of a scientific ambassador. I mean, okay, where are we going to find our information on these developing technologies? Scientific writing? Most people do not want to read that, trust me. So the news. But the news may not provide all of the information, or what is provided is often overly simplified. Now, the DNA barcodes that I showed you seem fairly simple, but those are just the beginning. These technologies are constantly being developed, and there's a technology out there now that takes DNA left behind from plants and animals called environmental DNA, or eDNA for short, and helps us track and monitor these species without endangering the researcher or the animal by trying to capture it alive. With environmental DNA, we can try to determine if Asian carp might be present in a water body or if an endangered species might be nearby without actually having to see it. When I was in grad school, I listened to one researcher's project on environmental DNA. He was trying to determine if bait shops that sold bait fish were unintentionally selling species of fish like Asian carp, thinking that they were just regular minnows. So he went to shops across the state of Michigan and he sampled water and used this environmental DNA method to determine what species were actually being sold. Now, it turns out that there were a fair amount of shops where the species, or at least some of the species being sold, were not the fish intended for sale. Luckily, very few of these shops, if any, had any evidence for Asian carp. But the point is that this researcher took one potential source of invasion, the sale of bait fish, and assessed the risk of introducing Asian carp into our waterways using nothing but samples of water. Now, at this point, this work is conducted by researchers, but there will come a day where anyone can do what this gentleman did using handheld, consumer-friendly genetic instruments. As technologies like environmental DNA and basic DNA processing improve, tools like these will help anyone determine if what's for sale is really what the seller claims, or if your dog has an infection and needs to go to the vet, or maybe even if the mosquitoes in your yard carry the Zika virus, all in a matter of minutes. It's going to be awesome. Now, there's another technology out there that you may have heard of in the news. It's a DNA editing technology referred to as CRISPR. With CRISPR, we can edit, add, and even remove genes from an individual's DNA. Just think that in the near future, we can engineer malaria-resistant mosquitoes, we may already be doing that, or remove the genetic source for Huntington's or Alzheimer's disease completely. The possibilities for CRISPR seem endless, but gene interactions are so complex that there is still a lot of work to be done, both technical and ethical, before we can fully understand the potential problems that arise when you alter the genetic foundation of any organism. Jurassic Park was a great movie, but we do not want the reality. And technologies like CRISPR and eDNA are really cool and they're really interesting, but there are drawbacks and limitations that might not make it into the news but are real and important nevertheless. And it's in instances like these where I can meet with people and help translate the science into information that can be used and understood by anybody. These technologies, as I've said, are constantly developing, they're constantly improving, and they will eventually play a role in just about every aspect of life, from invasive species control to public health, maybe even to what color we want our children's eyes to be. When these technologies will inevitably impact you, it might sound a little cliche, but knowledge becomes power. It'll become important to ask questions and keep our eyes peeled as more information comes into the news. In fact, there are people out there, like folks who work for university extension offices, whose jobs include educating the public and helping non-specialists understand this new information as it becomes more publicized. All you have to do is just do a Google search on extension offices in your state, and boom, there they are. Anyone can and should contact these offices or folks like myself who have developed services for the community based in this technology and ask questions. What is this technology? What can it do? What can't it do yet? And maybe most importantly, how do you know? Then we can really begin to understand what's being developed, and we could even get involved. There are programs out there now that turn anyone who's interested into citizen scientists. Programs like Eterna, Zooniverse, SciStarter, the Midwest Invasive Species Information Network, aka MISIN, just to name a few, turn research into games and crowdsourcing events that allow anybody to get involved with projects ranging from tracking wild animals to engineering, tackling climate change, even to studying DNA for medical research. Eterna, for example, has a puzzle game where players can essentially create variations of DNA to help researchers push forward with medical research. They are currently working on tuberculosis, and my friends and I are quite addicted to these games. MISIN, on the other hand, has an app where you take a photo of an invasive species and you can log that photo along with your location to a regional database to aid in the tracking and monitoring of invasive species. It's just point and click. So regardless of how little or how much effort you want to put into research, there's probably a program out there. If you take nothing else from this talk, please remember that while genetics as a scientific field and development of these technologies is conducted by specialists, that doesn't mean that the ability to contribute and the technologies produced have to be thought of as some science off in the distance that's beyond our understanding. We can all contribute, and we can all benefit. Not even a month ago, I went for a walk with my mother along the lake by my grandparents' house, and we noticed two things. One, that there were very, very few zebra mussels around. And two, that for the first time in 20 years, we saw clamshells from clams living in the lake. I thought our lake was changed forever when the zebra mussels took over. But in that moment, I felt this rush of hope that maybe, just maybe, our squirt guns have finally returned. My name is Syndell, and I thank you for listening.